Without further ado, I guess we'll do our first talk. And again, that's TEVAR acutely increases left ventricular work in an animal model by Dr. Stanko from Johns Hopkins.
Good morning. My name is David. Thanks to the VESS program committee for putting this on and for the moderators for being here this morning. Today I will be talking about how TEVAR increases left ventricular work in an animal model. Specifically, we use pressure volume sensing catheters and PV loop analysis to examine the acute effect of TEVAR on the heart. These are our disclosures. So this is a concept that everyone here is probably familiar with, the idea that you're in the clinic and you recognize a clinical problem, or you read a paper that's been published with clinical data, you have a problem that needs to be solved and needs to be innovated, then you go to the lab or the computer simulation or the basic science lab or translational lab and develop some innovation that solves that problem. This is true of probably every project that's being presented. It's a little different here because a lot of the clinical data has actually not been done by our lab. These aren't necessarily observations that are first done by our lab, and probably there's a bunch of people in the audience today who have made these observations, published these papers regarding the long-term effects, the medical effects on the heart of TEVAR and proximal aortic stenting. In fact, one paper that's not from our lab, but that's really well done in the Annals of Surgery about 1.5 years or 2 years ago, comes from, again, probably authors that are in the audience, and their data basically showed that in young trauma patients that got TEVAR for trauma, that there was chronic remodeling in the heart over the first 5 years. These are human patients that they followed, and it affected the LV mass, meaning essentially the mass of the left ventricle, as well as decrease, obviously was associated with decreased aortic compliance because you're placing a prosthesis in the aorta, and was associated with hypertension, heart failure, and a whole bunch of other effects that occur in the time period following TEVAR, especially in these young trauma patients. It's also been shown that there's chronic remodeling that happens in proximal aortic stenting from the chronic aneurysm literature, as well as from the cardiac literature in the ascending aorta. So there's a whole bunch of data out there that suggests that because of the effect of the compliance changes of the TEVAR stent, the heart has to work harder in order to overcome the resistance that increases in the aorta. And you think about it, that makes perfect sense, because as the heart squeezes, that systolic ejection is transmitted in kinetic energy to the aorta, which then has to be received during diastole. The aorta responds to that, relaxes, and then has to accommodate during diastole. So if you place a stent in the aorta, it makes perfect sense that the compliance overall decreases and the resistance in the vascular circuit increases. So we wanted to study this and see what happens, specifically what happens immediately after TEVAR in the minutes that follow TEVAR in the acute setting, because to our knowledge, no one's looked at this. We use swine because they're a great model for this project, because their aortas, we do 50 to 70 kilogram swine, so you can use human stent grafts that are commercially available and everybody has access to, and you can place them in the aorta the same, with some minor modifications in the way that you would a human TEVAR system. So a little bit of background on the physiology. So this is something that everybody's familiar with, this sort of idea that you have a pressure tracing in the left ventricle over time. You also have a volume tracing in the left ventricle over time. Those two things are coupled, so as the volume changes, the pressure changes. You can express that by removing time and thinking about the cardiac cycle. So this is one cardiac cycle with left ventricle pressure over left ventricular volume as what's called a pressure volume loop. And from this, you can get gold standard biomechanical information on the left ventricle. For example, you can tell, I don't have a pointer, but for example, the difference between the end diastolic volume and the end systolic volume is the stroke volume. The area inside the curve is the stroke work. The point at the top left that's labeled number 3 is when the aortic valve closes, and the pressure there is the end systolic pressure, and the volume there is the end systolic volume. So you can get load dependent variables, meaning that depending on what your preload and afterload is, these things change. You can also generate those red lines, which are load independent parameters, meaning as you change your preload and your afterload, your end systolic pressure volume relationship should be maintained the same, assuming that you don't change the ionotropic effects of the heart. Same thing with the end diastolic pressure volume relationship, which is the red curve along the bottom. So basically the curve can migrate within this sort of boundaries of those red lines, assuming you don't change the contractility of the heart just by changing the preload or afterload that the heart's seeing, particularly the left ventricle. This is how we actually do it in the pig, is we have a pressure volume sensing catheter. So this catheter you see in the left ventricle there has a pressure manometer at the tip, pressure sensing manometer at the tip, and it can also sense the volume. Probably not going to get into that right now, but the idea is that you can tell in real time what the left ventricular pressure and the left ventricular volume are in real time. And we can do this all endovascularly. This is on the left is just an X-ray showing the percutaneously placed left ventricular PV sensing loop. Can you hit play on that video? Is that possible? On the right? Since I don't have a mouse. This is a left ventricular pressure volume catheter you can see there in the apex of the heart during an IVC balloon occlusion. So during this video, we're essentially inflating a balloon that's in the IVC and dropping the preload significantly. So if you remember from that last slide, those red lines where you're changing, the PV loop is going to migrate within the boundaries of those red lines. We'll come back to this, but I wanted to show this here. Obviously, if you do this to a pig over and over again during an experiment to get those load-independent parameters, it causes a relatively large physiologic effect on the animal because you're inflating a balloon in the IVC, you're dropping preload. They have a response to that. It can take some time for them to recover. Oh, and this is just an oblique view of the same. Oh, okay, that worked. So trial timeline, so basically we instrument the pig, we get vascular access, we place a LV pressure volume sensing catheter, we obtain this baseline cardiac data, we then TEVAR the animal, and then we obtain post-TEVAR cardiac data to essentially compare the pre and post left ventricular function. We did 6 swine, one of which underwent IVC occlusion. Again, we did that as sort of a sensitivity analysis because it can cause, again, a relatively large physiologic effect on the heart. So this is our results. So the first bit is that the end systolic pressure, which is, again, on the bottom of the PV loop, the change that we see over time, there was no real change in the end systolic pressure before and after TEVAR, which is somewhat expected because this is, again, in the left ventricle, and there would be no reason that we would expect that there would be a change in the end systolic pressure at that time. The end systolic pressure, however, did increase in the left ventricle before and after the TEVAR, which makes sense if you think about it because, again, you're increasing the decreasing the aortic compliance, you're increasing the amount of work that the left ventricle has to do to generate the same systolic pressure, and in doing so, by increasing the vascular resistance, you're affecting you're increasing the end systolic pressure before and after. So this is an extra piece of data that says that in addition to all all the chronic data that we're seeing over and over again in the literature, that there's actually an effect in the left ventricle immediately after the TEVAR that isn't necessarily seen on the chronic data or maybe has mechanistic information. And this is just what this actually looks like in the lab. So the purple line on the first one's the EKG. The second one is the pressure tracing in the left ventricle. And you can see that the area that's selected is the pressure volume tracing on the right-hand side of the screen. So as we inflate the balloon in the IVC, the preload drops off, the pressure drops off in the LV, and the pressure volume loop goes like this and migrates down into the left lower quadrant. And then we can draw the end systolic pressure volume relationship, the end diastolic pressure volume relationship, and determine those load independent parameters. So that's how that works. And this is what it actually looks like in the lab. And if we do that before and after the TEVAR in the same way, you can see that, again, we repeat that. We do that once, then we do the TEVAR, then we do that again. Then you can see that the end systolic pressure volume relationship actually migrated, which means that there's actually an ionotropic effect or contractility effect in the heart that required in order to generate the same sort of global physiologic state. We also saw that the end IVC occlusion potential energy migrated. We're not entirely sure clinically how relevant that is, but we're looking into that now. And the end diastolic pressure volume relationship remained unchanged. Again, so this is a sort of you might say more sophisticated way of, in this one animal, of supporting the data seen in the previous 5, which is that there seems to be a change to the end systolic pressure volume relationship, but not necessarily the end diastolic pressure volume relationship. So overall, the point here is that TEVAR was associated with an acute increase in LV end systolic pressure and a shift in the end systolic pressure volume relationship, and this together indicates that there's increased ventricular work in the left ventricle, and this may provide some sort of mechanistic insight into how patients develop hypertension, heart failure after TEVAR, and then also what we can do to generate next generation stents, which theoretically, based on this, you'd want to have the compliance of the stent match the compliance of the aorta, so that when you introduce the prosthesis, it doesn't change the overall global vascular resistance of the reservoir, and hopefully this won't happen. Thank you.
University of South Carolina. Thank you for helping us out with changing up some of the schedule to accommodate travel issues. I do want to thank you for that. And very nice presentation. I just was curious if you guys had plans to look at this over a longer term, since here you were only looking at the changes immediately after deployment. It isn't able to capture any additional compensation that may happen over a longer span of time.
Yeah, that's a great question. And one of the best thing about the pressure volume sensing catheter system is that it's basically gold standard, left ventricular biomechanical. You can get those gold standard parameters from it. The downside is that it's highly invasive and you have to put the animal back under general anesthesia and reintroduce this pretty, quite frankly, it's like pretty stiff, 5 French wire is basically what it is into the left ventricle. And so in order to do that, it's pretty invasive. Obviously, you can put an echo in the heart and you can get those parameters intermittently if you'd like. And we are sort of planning on a longer sort of acute study where we'd follow them for hours instead of just looking sort of immediately pre and post TEVAR.
Our hope is to work up to months, but sourcing money for this, is part of that challenge. So we take PayPal, cash, all those things.
Changing to a survival lab certainly makes a big difference. Financially.
And here we were recovering the stents, cleaning them, and then repackaging them and using them again. So it would involve a kind of big step change in cost, but that's absolutely the plan.
Jim McKenzie, New York. Fascinating work. I think this is something we all need to see more about, and I applaud you for taking the time to get the acute data, then you can expand it with more chronic data. Going back to your model, how much do you oversize your stent graft? And the majority of stent grafts are actually put in aneurysmal disease, and it's much smaller percent for transection, which would be more towards your model. And I guess my question is, if you have 2 to 3 centimeters constrained, but then the majority of the stent graft in an aneurysm, how does that impact on your model where the graft has more compliance to move than if it's constrained? And again, how much you oversize? But great presentation. Thank you.
Yeah, really good question. So one of the things we want to do when we do that next study is use atherogenic pig model, where basically we overfeed some Yucatan mini pigs and give them atherosclerosis and build a aneurysm model, a surgical aneurysm model that way. We haven't done any of that data yet, but we hope to. So to answer your second question, I'm not entirely sure how that would play out. My guess is that that a chronically sort of conditioned patient, the effect on their heart may be less significant than it would be in a sort of a normal heart, normal aorta, young trauma model that we sort of do here. But I think that's super interesting. And then the second bit is we actually do the opposite. So Medtronic was gracious enough to donate some expired stents. So we had a whole bunch of 20 millimeter diameter stents. So we actually ordered the pigs and sized the pigs to the stent, and then we put them in our CT scanner to make sure they were the right size. So we actually kind of did it backwards in order to save money. And we oversized them about 15, 20%. Oh, yes, exactly. Dr. McGee?
Hi, very nice presentation. I'm just curious, probably you had too small numbers to consider doing this, but I'm curious whether you notice any observations, whether you put a single, a second, or even a third stent graft. The length of coverage might be interesting because you're just measuring this with echo, presumably during the procedure, but you could have taken the 6 pigs and made 18 pigs out of it by doing it in the...
Yeah, we actually did that on a few of the pigs. We stented basically did the normal TEVAR, and then we extended down to the celiac, and then we stented between the celiac and the SMA, and then we stented some all the way down to their iliacs. The problem with that is, well, first off, the first observation is that we noticed what you thought, which is that basically there's a bigger difference on the proximal aorta, so that's why we focused on that, because it seems like, for example, EVAR versus TEVAR, for example, there's a pretty big difference in the physiologic effect from the TEVAR versus the EVAR. The other thing is that if you do we try to do serial balloon IVC occlusions, and the animals just do not tolerate like five IVC occlusions in a half an hour, 45 minutes, or whatever it is. So that's the downside there. So I think we focused on this to sort of work out the model, and now I think we're fully equipped to do that. Thank you. And